Okay, so the first one that hopefully everyone knows about is green fluorescent protein. This originally comes out of a jellyfish. And in 2008, there were three winners who won the Nobel Prize in chemistry for developing the fluorescent protein pellet. So Shimomura was the person who first identified the GFP molecule and started to do the characterization. Martin Chalfie was the person who first took that and attached it to a protein of interest and started to actually use the GFP molecule as a tracker for proteins within cells. And then Roger Chen took the GFP and found new ways of creating a much larger color palette. So now we have a full visible light spectrum, essentially, of fluorescent proteins that are available to us. Okay, so important things about expressing fluorescent proteins. So this requires genetic modification. Normally you're creating plasmids or integrating into a genome that's creating a fusion of the green fluorescent protein signal with your protein of interest. So POI here is protein of interest. There are two ways that you can attach that. So you can attach the GFP molecule at the C terminus of your protein of interest, or obviously you can attach that at the N terminus as well. There are a few considerations that you need to think of before you start designing your fusion proteins. So very often proteins have trafficking motifs that may appear either at the N or the C terminus of the protein of interest. So obviously you need to be aware of those, that you're not getting in the way of any of these motifs that might cause aberrant trafficking of the protein. One thing is the secondary structure. So if your C terminus is embedded deep within your molecule, if you're attaching a 27 kilodalton GFP molecule to that, then that is obviously going to mess around with the secondary structure of the molecule that you're interested in, or the protein that you're interested in. So again, that could cause aberrant trafficking of the protein that you're interested in. The final one is the expression level. So very often, if you're using transfection of a plasmid into cells, for example, then they'll be under control of a certain promoter. Very often these are viral promoters, which are very, very strong. And as the figure shows you on the bottom here, it's best to have as low as an expression as possible. If you start to overexpress, then again, you can perturb the system that you're trying to look at, and that's never a good idea in terms of trying to research something if you're actually changing how it behaves. So these are kind of key considerations to think about before you start planning your fusion protein experiments. Having said that, GFP and other fluorescent proteins are widely used now across numbers of different model species. So we've got bacteria, worms, flies, rabbits, mice, whatever application you need, really. Just going back to the GFP molecule itself. So as mentioned, it's a genetically encoded protein. So it's built up of a string of amino acids, and this folds into what's called a beta barrel conformation. So it has these beta sheets, which form the barrel around the outside. And in the middle, the fluorophore is generated from 3 amino acids. So this tyrosine 66, serine 65, and glycine 67. And this highlighted in green here is the fluorophore section of that molecule. As I mentioned, Roger Chen really pioneered the development of new fluorescent proteins. So we started off with the green, but you can see if you go into the GFP molecule and make these mutations, then we start to open up a completely different color palette. So we've got blue fluorescent proteins, cyan, yellow, sapphires, obviously in the red. And if we look at a different molecule, so the DS red fluorescent protein, again, lots of researchers have gone in and made specific point mutations here, which again have created a new groups of fluorescent proteins that can be used. So the cherry that you may have heard of, D-tomato, so monomeric proteins as well. So now we've really got to a point where you can go from the blue range all the way out into the kind of deep red range using different fluorescent proteins. Just to extend the properties for fluorescent proteins here, so these are the same, so these will have the same properties that we had just for our fluorescence molecules. The two at the bottom here that I've added specifically for the fluorescent proteins, and I've touched on it already, is the oligomeric state. So a lot of these molecules like to dimerize. So a lot of work has gone into trying to create monomeric versions of these fluorescent proteins. So again, this is a key consideration. If you're tagging your protein of interest with a molecule, you want to make sure that that's not going to dimerize and again change the behavior of the molecule you're investigating. The other thing to take into consideration is because it's an amino acid, it has to fold correctly, and also the fluorophore has to form from these three molecules. So it can take a bit of time for the fluorescent protein to mature and get to a state where it can actually create fluorescence. So again, a consideration in terms of setting up your experiment. There are now hundreds of fluorescent proteins available, so if you are confused about which ones you should use, either speak to your local facility, come and speak to us if you have any questions. You're based in Melbourne, or if you're somewhere else, go and speak to your facility there. Otherwise, there's a very good website, fpviz.org, which allows you to select fluorescent proteins based on a number of different parameters. And basically determine which one is best for the experiment that you want to set up. So a lot of these are these fluorescence properties that we've discussed in the last couple of slides. The nice thing about having that full visible spectrum of fluorescent proteins is we can do some co-staining experiments. So this is just an example taken from molecular probes. So Thermo Fisher website of a commercially available kit where we have two independent plasmids which are expressing one GFP and one RFP, and these are going to different compartments, or can go to different compartments within the cell. How do they do that? Well, I mentioned trafficking motifs earlier on. They can make use of those. So if you want your fluorescent protein to go to the nucleus and stain that, you can use a nuclear localization signal. If you want it in the cytoplasm, you can use a nuclear export signal. If you want it to go to the endoplasmic reticulum, then there's an ER retention signal, which is a KDEL motif. Or you can tag it to known proteins that go to certain compartments. So the pyruvate dehydrogenase complex goes to the mitochondria. You've got LAMP1 that goes to lysosome, RAB5A that goes to endosomes. The list goes on depending on what structure, what organelle you're interested in the cell. So you could tag your protein of interest, say in this case in the YFP channel, and then use a mitochondrial RFP as a marker for the mitochondria, and you can then start to have a look at whether or not your protein is going to where you're expecting it to go to. One of the fun things you can do if you're bored on a Friday afternoon and you have access to them all. So this is a petri dish with bacteria expressing fluorescent proteins of different colors. So you can have your own little art project. This is actually out of the Chen lab. So this is an example of the different colors of fluorescent proteins that they have available.